Hello, hello. Welcome back. This is going to be chapter 3, which covers cell signaling and endocrine regulation. One of the concepts that chapter 1 tackled was the need for organisms to have homeostasis, internal consistency. In order for this to be successful, cell communication is going to be essential. And this will allow the organism not only to communicate internally, but also receive output, or I should say input, from external stimuli, thereby being able to regulate things like its temperature and its internal pH. What chapter 3 is going to do is it's going to go ahead and take a look at all the different cell types of communication and signaling that we can be utilized. And we're going to compare and contrast them. And then towards the end of the chapter, we'll take a look at some specific hormones that we have across different types of animal species. But before we get towards that, let's go ahead and take a look at this slide, which compares the difference between direct and indirect cell signaling. Now, direct cell signaling usually means that you're going to communicate from one cell to another, and it usually involves the lining up of the cell membranes and utilization of what's called a gap junction. Now, in my little drawing, I'm making a little tunnel that's going to connect my two cell membranes, and this is going to represent my gap junction. It almost looks like a little hole punch that's being made, and what's nice is that it allows for cytoplasmic content to be shared from one cell to the next. So you can have your sodium, your potassium, your chloride, you can have those ions flowing in and out. And we see, for instance, that it's very popular among cells that are electrically stimulated, like, for instance, your myocytes, your heart cells, as well as your muscle cells will have high amounts of gap junctions that allow the cells to directly communicate. On the other hand, if you want to do more of a long-term messenger signal, meaning that maybe you want to communicate from one organ to the next, or even to a cell that's not directly close to you, you're going to have to utilize indirect cell signaling. This is going to involve the release of a chemical messenger, such as a hormone. The cell will release the hormone, and it will often travel in the bloodstream or any type of extracellular fluids. And what we see happening is that it will need to connect with a target cell before the cell signaling pathway can be completed. The target cell will respond to the chemical messenger and will often induce things such as activating a transduction pathway, maybe that it will go ahead and increase the permeability of the cell membrane, maybe it will adjust the voltage membrane, so it will induce depolarization or even repolarization to occur, or you might even just have a cascading effect of secondary messengers that might be activated based on the interaction between the messenger and the target cell. Either way, it's important to note that when you do indirect cell communication, that your messenger has to connect with the target cell before you can get your response. And when you're doing your indirect signaling and you're releasing these chemical messengers, we see that you can often either go a short distance or a long distance in your travels. The short distance usually will entail the fact that you will have what we call a paracrine or an autocrine secretion. Now, paracrine means that the chemical messenger will find its target cell in a very nearby cell. Oftentimes, this will be within the same organ that it was secreted from. So, for instance, the pancreas, which many of us know secretes insulin, also secretes another hormone called somatostatin. Somatostatin is secreted by the pancreas, and its target cells are located in the pancreas. Because of that, that would be an example of a paracrine secretion. If we see that the messenger actually will be diffused and signals back to the original cell, meaning that it stays with a direct contact, so the chemical messenger is diffused back to the signaling cell, aka the signaling cell will become your target cell, then we call that an autocrine secretion. And auto means self. So this is, for instance, what we see with the interleukins that are secreted by certain monocytes. The monocyte will secrete the interleukin, and then the interleukin will bind to a receptor on that same exact monocyte. So that would be an example of an autocrine secretion. Most of the time when you do your indirect signaling, though, you're traveling more of a longer distance. So in that case, you want to take a look at things like what type of transport are you using, what is your end product, and that will allow you to divide it between the endocrine system and the exocrine system, as well as the nervous system. So over here, what we see happening is the mention of the endocrine system, which utilizes hormones as they're being transported through the circulatory system on their way to find the target cell. Another way that you can do your long distance is to utilize your good old-fashioned nervous system, right? Use the neurons within the brain and the spinal cord. And what we see happening with the nervous system is that you'll often depend on a neurotransmitter to be released, and you'll rely on synaptic connections between the neurons as well as their target cells to commensurate that message. So here is an illustration of just a few of the cell signaling. In A, we have a nice representation, definitely better than my drawing, of the gap junctions. So notice how they create a pore that allows cytoplasmic content to flow directly from one cell into the next. B is going to take a look at autocrine and paracrine secretion. So this will fall under indirect communication, the short distance one. And one thing that I want to point out to you is that you're going to notice that you have your chemical messenger that's being released. And we can see it has two options. It can either turn back and respond to a receptor that's located on the same signaling cell. In that case, that would be an example of autocrine. Or it can go ahead and diffuse a little bit down the road and make a receptor connection with a very closely related target cell, most of the time still within the same organ. And in that case, it would then be an example of a paracrine secretion. Now, in case you're wondering, well, how does this differ from an endocrine signaling? Well, remember, with the endocrine, we are also releasing our chemical messenger, but we have to utilize the circulatory system to travel. So there's much more of an elongated pathway before the chemical messenger can actually go over to the receptor on the target cell. The target cell will not be the same as the signaling cell, and it will also not be located within the same organ system. And then last but not least in our illustration, we have a beautiful neuron right over here indicating the use of the nervous system. And in this case, what we're doing is that our neuron becomes our signaling cell, and it can go ahead and send off electrical signals and cause the release of neurotransmitters so that it can interact with the receptor once again on the target cell. So lots of variations to choose from. It all depends on the distance that you're traveling and what the ultimate goal will be of activating the target cell. Now, to stick on the topic of direct signaling, I just want to show you a little bit more of a detailed picture of your gap junction. So I mentioned to you before, it is you allowing to share cytoplasmic content between neighboring cells. And what's really cool about this is that the actual gap, the opening, that one's going to be allowed to be made by these interlocking proteins that are called connexin. Now, on your PowerPoint, you're going to notice that it says connexin, and then in parentheses, it says or inexin. Inexin is what the protein is called when we take a look at invertebra samples, because we have seen gap junctions in both vertebra as well as invertebrate muscles. Now, the nice thing about a gap junction is that it really allows the movement of ions to seamlessly go from one cell to the next. So it's an excellent way to change the membrane potential, especially if you have an excitable cell that can easily depolarize and reach action potentials. You can also utilize it for repolarization, which would often mean that you're resetting the cell back to its resting membrane potential. We also know that you can have chemical messengers that can travel through the gap junction. One of those that's extensively studied is your CM, or your cyclic AMP. We know that it's able to activate some of the secondary messengers along the way. So if you bear with me as we proceed through our chapter, we're going to go ahead and visit our cyclic AMP or CAMP a little bit more detail when we get to those particular slides. The gap junctions can also open and close. They can be regulated, all in the essence to maintain the ion flow. So for instance, it's quite common that if the extracellular content of calcium gets too high, some of the gap junctions tend to close to prevent any additional calcium from flowing in. On the other hand, if it sees that the internal pH is becoming more acidic and moving away from a neutral environment, oftentimes there'll be an increase in the opening of the gap junctions to allow your hydrogens to flow in and out as to regulate the internal pH of the cell. When it comes to our indirect signaling, when we're having to travel some of the distances, what we see happening is that you're going to involve 3 steps. And this is just a really quick overview. We'll be looking at much more detailed examples as we proceed to the chapter. But one thing that you're going to definitely need is you're going to need to have a signaling cell. The signaling cell, which can be part of a gland, it can be part of an organ, will be the one that will be actually secreting the chemical messenger, such as your hormone. Then what we see happening is that we need to have some type of transport mechanism so that we can reach our target cell. And more importantly, when we get to the target cell, we see that oftentimes it will require specific receptors before it can actually cause an activation pathway to be stimulated. I do want to take a few seconds out and I want to talk to you guys about the difference between the endocrine and exocrine system, because both of them can be used for indirect signaling. Alrighty, so here we have an illustration that is going to go ahead and compare the difference between an exocrine gland and an endocrine gland. So let's go ahead and start with A. When you have an exocrine gland, what we see happening is that any of the secretions that are produced by the gland are usually transported in a duct or a tunnel system. They do not need to rely on the circulatory system. And when the secretion gets over to its target cell, usually it's located in what we like to call an open environment. So it's secreted into a cavity or on the external environment. So for instance, I'll give you an example. Sweat, mucus, digestive enzymes. Those are all examples of exocrine secretions. So for instance, when you sweat, what we see happening is that the sweat will travel through the ducts and it will be released through the pores out of the skin, allowing the body to cool down. When you have mucus, we see that the glands will go ahead and secrete mucus. It will travel into a duct and it's often released into the cavity, like maybe the internal lining of the stomach or the internal lining of the small intestines, or even within your saliva, you have mucus that's being secreted. The buccal cavity is a cavity, so it's an external opening where it's being secreted to. What else? The digestive enzymes. Your pancreas produces a boat of different pancreatic enzymes that can be used to break down proteins, lipids, carbohydrates. When these enzymes are produced by the acinar cells in the pancreas, they will travel through the pancreatic duct and they will be released within the open cavity of the duodenum, which is the top part of the small intestines. So anytime you see that you have a secretion that has to travel through a duct, not the blood system, and it's basically secreted into an open environment, we're looking at an exocrine secretion. On the other hand, if we come over to endocrine, then with endocrines is when we're talking about good old-fashioned hormones. So we're talking about our chemical messengers that are released by our secretory cells. They have to travel in the circulatory system, and they are not fully active until they meet the receptors of their target cells. So an exocrine secretion is one that is released into the bloodstream and that will have to connect with the receptors of the target cells. Now, is it possible for a gland to have both exocrine and endocrine capabilities? The answer is yes. And actually, there's a bunch of different examples for mixed glands. I think we could probably start with the pancreas, since we've mentioned the pancreas already about the digestive enzymes. So the production and the secretion of the digestion enzymes are an example of the exocrine secretion for the pancreas. On the other hand, if we think about the pancreas, we also think about hormones such as insulin. When insulin is produced by the pancreas, it is dropped into the bloodstream and it circulates the body looking for its target cells. That secretion would be an example of the endocrine gland capability of your pancreas. How about your reproductive organs? So for instance, what if we take a look at the testes? What do the testes produce? Hopefully right now you're answering me back and you're going sperm, 100%. So testes are able to produce sperm. Sperm is dropped into the vas deferens. It will travel through many places, including the penis, and eventually it will be ejaculated out. So that would be an example of an exocrine gland. It had a duct and it had an external cavity that it was released into. On the other hand, if you think about your testes, what hormone comes to mind? Testosterone? Correct. Testosterone will be released in the bloodstream and will then travel the body looking for its target cell. So that would be the endocrine aspect of the testes. Ladies, don't feel left out because our ovaries do the same exact thing. Ovaries are another example of a mixed gland in the fact that ovaries are able to secrete an oocyte, an egg. It will travel through the fallopian tube, there's your duct, and it will either empty out into the uterus, which is your open cavity, or in the case of the menstrual cycle, it would be expelled out of the body, also an open surface. So the oocyte is your exocrine secretion, whereas the estrogen that you produce that goes into your bloodstream and reaches the target cells, that would be your example of your endocrine secretion. So when you're doing your quiz or when you're reading your questions for your exam and we're talking about signaling and indirect signaling, make sure to read if we're using a duct or the bloodstream, if you're going to an open cavity or to a target cell, because that is going to help you distinguish between an exocrine and an endocrine secretion. I also want to show you this table. I really like this table because what it does is it gives you a really nice way to compare your autocrine, your paracrine, your nervous system, as well as your endocrine and exocrine secretions. It's a fantastic way to kind of keep in track of what's happening. And one thing that you want to kind of keep on mind on is basically things like what are the method of transport, what are the speed, and how long is the response. And if we come over here for the nervous system, for instance, let's compare nervous system to the endocrine system. In the nervous system, we see that our transport has to occur along this axon. So it travels through the axon and it will eventually make the synapse. The synapse is the connection point where it either meets with another neuron or, for instance, in the case of the neuromuscular junction, where it meets with the skeletal muscle. On the other hand, look at the transport for the endocrine system. The circulatory system, there you go. How about speed? It turns out that the nervous system can have a very rapid response, especially through the fact that axons tend to be myelinated, so it tends to speed up the nerve signal. On the other hand, the endocrine system tends to have a more slow type of response. And part of that, slow type of speed. Part of that is because it has to be released into the circulatory system and it has to travel its way all the way over to the receptors. So what does that mean for the duration of the response? With the nervous system, as soon as you take the stimulus away, the response goes away as well. So you tend to have a very short duration. Whereas with the endocrine system, even though it was a little slow in responding because of the method of traveling in the circulatory system, as long as you have free-floating hormone, you can have a response when it makes a contact with the target cell. So it tends to have a longer duration. So as you can see, as you peruse through this table, each category has a benefit and it has a drawback. It really all depends on what the ultimate goal is for cell communication. The next item that I want to explore with you are basically from a chemical standpoint, what separates certain messengers from each other. And the reason this is important to bring up is because it's going to determine if our messengers are going to be able to be classified as hydrophilic or hydrophobic, which will thereby also affect how the target cells will interact with these chemical messengers. So on your screen, you can see that all chemical messengers, whether we're looking at paracrines, autocrine, exocrine, endocrine, they basically fall in 6 categories. And within those 6 categories, the ones that we're really interested in are the ones that are going to be able to give us our endocrine secretion and make our hormones. And those are going to be the peptides, the steroids, and the amines. Now, what I want to do with you guys is I want to go ahead and take some time and talk about these 3 categories, and we'll talk about how they interact with the target cell and if it is a rapid or a more slowed down response that they will initiate. Alrighty, oh, before we get to that, my apologies. So here's a table that will show you the difference between a hydrophilic and a hydrophobic messenger. Now, what you're going to see is that if your hormone is peptide-based or amino-based, it's going to be a hydrophilic hormone. And hydrophilic means that it's water-loving, and because it's water-loving, that means that your body can go ahead and produce it in bulk. It can store it in extracellular vesicles. When it's needed, it has already been produced, so usually you're able to release it quickly. It can freely float within the extracellular fluids. So within the blood, it doesn't have any issues with that. And when it gets to the receptor cell, it's not able to pass through the cell membrane, so it will have to interact with the transmembrane receptors, meaning receptors that are located on the plasma membrane of the target cell. That's a very important point to kind of highlight, the fact that hydrophilic messengers are not able to enter through the cell membrane due to its phospholipid properties. So therefore, the receptors will all be located transmembrane, so on the surface of the cell membrane. Hydrophobic messengers, on the other hand, are water-fearing. And since our body is mostly water, both intracellular and extracellular, what we see happening is that these messengers, which by the way will be based on a chemical standpoint of being composed of steroids, those hydrophobic messengers will be synthesized on demand. They will also have an easy way of diffusing across the membrane because they can interact fully with the cell membrane. So they'll diffuse out. They do not need exocytosis. If they do make contact with the extracellular fluid of the blood, oftentimes we see that they'll utilize a carrier protein to protect them in longer distances. If it's a short distance, usually they'll try to travel by themselves. But either way, when the hydrophobic messenger gets over to the target cell, usually what we see happening is that they'll have a choice of entering and connecting with an intracellular receptor or staying outside and utilizing the transmembrane receptors, just as the hydrophilic messengers will do. And I'll tell you, many of them will not choose that second option. A lot of hydrophobic messengers will go ahead and enter into the receptor cell and will make contact with intercellular receptors. And these receptors are usually located on the nuclear envelope, so by the nucleus. Now, when they interact with their intracellular receptors, they tend to have a slower effect. And the reason for that is because oftentimes that will activate a genomic change. And the fact that it has to look for the certain gene and it has to initiate transcription and translation, that'll take a little bit longer for you to get to your ultimate effect. On the other hand, and I don't think I'd mentioned this to you before, with your hydrophobic messenger, or I should really say any messenger that's able to bind through the transmembrane program, what we see happening is that these receptors will often have a very rapid response. And that's because they're able to initiate a cascading pathway that will involve a lot more secondary messengers, thereby amplifying their response very quickly. They do not need to initiate a genomic change, unlike what we see in the intracellular receptors. Alrighty, so here's a look at our peptide-based hormones. As you can see from the name, it is composed of amino acids. When we take a look at where it originates, it turns out that we rely on the ribosomes and the rough ER. And what's going to happen is initially it will come in the form of what we like to call a pre-pro hormone. And a pre-pro hormone usually has multiple copies of our peptide hormone that are replicated and attached to each other. They will also have a signaling sequence that can be utilized to target secretion. And what we need to do is we need to take this pre-pro hormone and we need to start cleaving it and getting it into its final active form. So what we see happening, and I'll show you this in an illustration in just 1 second, is that the pre-pro hormone will go ahead and be secreted by the rough ER and the ribosomes. It will then go ahead and travel, and right before it leaves the rough ER, it will be cleaved so that the signaling sequence is released, allowing for the pro hormone, because we're going to release the pre from the pro, allowing for the prohormone to travel over to the Golgi apparatus where it will be placed in little vesicles, little containers. The vesicles will often have enzymes that can go ahead and cleave the prohormone into its active form. And from that point on, what we see is that the vesicles will do exocytosis. They will exit the target cells. And because peptide hormones are hydrophilic, they can freely travel into the bloodstream until they get to their target cell. When they get to the target cell, they will bind, remember, to the transmembrane receptors. They do not enter the cell because they cannot pass through the plasma membrane. And because they bind to the transmembrane receptors, we see that they activate a signal transduction pathway that often activates secondary messengers and causes an amplifying response. So you'll quickly have an effect that is desired by your target cell. Let's take a look at this from an illustrated aspect. Okay, so here on the left-hand side, we have our rough ER, and the little red dots are the ribosomes, and we see that they're able to go ahead and generate our pre-pro hormone. Here we go. And within the pre-pro hormone, not only do we have multiple copies, but we also have our signal sequence that will activate it to be cleaved. So you can see it's being cleaved here. We remove the signal sequence, and then we start taking our copies of what we now are going to call a prohormone because we've cleaved off the pre-hormone, right? So now we have a prohormone, and this will travel over to the Golgi apparatus. And the Golgi apparatus will sort it, it will stack it, it will place it into a nice little vesicle. And within the vesicle, we see that we have different enzymes that can go ahead and cleave the pro of the hormone, allowing for the active hormone to be generated. And then it will be exocytosed out of the host cell. And there it goes into the bloodstream on its way to find its transmembrane receptors. And in fact, here we see a look at our transmembrane receptors. So this part right here, this is the cell membrane. This is the inside of the cell, and this is the outside of the cell, okay? So extracellular, and as you can see, the cytoplasm. And what this illustration is trying to show you is that our transmembrane receptors will have three domains. They will have the ligand binding domain. That gets its name because that's where the ligand or the chemical messenger, aka your hormone, is going to bind to. Then you have the transmembrane domain that will allow the signal that a ligand has bound from the outside. And then we have our intracellular domain, and the intracellular domain will often change its configuration, thereby indicating that it's been activated, so it can go ahead and cause a rapid change. So anything from, like, for instance, activating secondary messengers, or it can change the membrane potential, it can cause phosphorylation to occur. Either way, you'll have a rapid response on the target cell. Now, our steroid hormones are going to follow a slightly different path. And part of that is because steroid hormones are composed of cholesterol, so they are going to have more of a hydrophobic quality. They are synthesized by the smooth ER, as well as some of them are going to be produced by the mitochondria. We see within the endocrine system that we have three categories or classes that they can fall into. We can have ones like aldosterone, which are an example of a mineral corticoid. They are able to regulate our electrolyte balance, so things like our sodium, our potassium, as well as our water balance. Then you have the hormones that play a role in your stress response. Those are things like your cortisol, your cortisone, your corticosteroid. Those all fall under the glucocorticoids, and they will come into play when we start talking a little bit about our fight or flee response towards the end, or basically any time that your body goes through any type of stress. Last but not least, the third class of steroid hormones, those are going to be all our reproductive hormones that we're going to encounter. So when we talk about testosterone, when we talk about estrogen, progesterone, estradiol, all of these will play a role in reproductive systems, which will fall under the category of being a steroid-based hormone. I also added another hormone that we see in invertebrates, and that one is your ecdysteroids. It's right over here. This particular hormone, in addition to it being a steroid-based hormone, is very essential to invertebrates because it will help with the molding process. So if we're taking a look, for instance, like at arthropods, where they have to mold and release their exoskeleton, this is the hormone that will help them with that particular process. Now, because your steroid hormones are hydrophobic, their pathway to the target cell will be slightly different. The first difference that we're going to encounter is the fact that they're not produced in bulk, so they're produced on demand. The second one is that whenever they're ready to release the signaling cell, they can freely diffuse through the membrane. So they don't have to rely on the little vesicles, they don't have to do exocytosis, they're freely released. However, when they do get access to the bloodstream, we see that if they're going to be traveling a longer distance, they tend to have to piggyback on a carrier protein. So if you come over here to your illustration, you're going to notice that some of the hormones have been linked together to these little green items. Those are your messengers. So they'll link together, and when they're linked together, the carrier protein basically protects them from the water component found in the blood. This thereby increases its half-life, making it more likely that it will be able to reach its target cell. If the hormone has to travel a short distance, sometimes it might not rely on a carrier protein and it will just travel unbound through the blood system. However, it's obviously not recommended because of the hydrophobic components. Now, when the hormone gets over to its target cells, what we see happening is that it will have an option of either utilizing transmembrane receptors, and if it does so, it will have a rapid response, just like we saw on our previous slide when we were discussing our peptide-based hormones. Or what it will tend to do, and it tends to favor this is the number one choice of hydrophobic hormones, it tends to find an intracellular receptor. And the intracellular receptor means that it has to pass through the plasma membrane, which it can easily do because of its hydrophobic components. It will interact with a target that's inside the cell, usually near the nucleus, and when it does that, oftentimes it will go ahead and it will cause a transcriptional change to occur, or a genomic change. So as you can see on your PowerPoint, it says it has a slow effect on the target cell because it needs to activate gene transcription. Not all of the steroid hormones will do this. So for instance, cortisol tends to always utilize a transmembrane receptor, and because of that, it tends to have a more rapid response, a non-genomic based response compared to the other steroid hormones in the category. Our third class of hormones based on their chemical component are those that are based on amines. Now, amines are going to include the fact that they have an amine group, which is the nitrogen attached to the two hydrogens. Here you go. And as you can see, there's a wide variety of chemicals that fall in the amine group. Some of them are what we call neurotransmitters, so things like dopamine, for instance, whereas norepinephrine and epinephrine are technically both neurotransmitters and hormones. And then you have what we call our true hormones, like our serotonin and our melatonin, which help with our circadian rhythm. It helps regulate our day versus nighttime activity, when you're supposed to be active versus when you're supposed to be asleep. And then thyroid hormone, which helps with a whole bunch of different things, including your metabolic reactions and heat productions. Now, there's an interesting thing I have to tell you about thyroid hormone. Thyroid hormone starts off from an amine aspect, and that means that it is going to be hydrophilic. Because right over here, we know that anything's based on amino acid, just like our peptide hormones, are going to be hydrophilic. So thyroid hormone in its inactive form will go ahead and start off with that same quality, the fact that it is hydrophobic. However, what we do see happening is by the time the precursor hormone is cleaved into the active form of thyroid hormone, we see that the quality has changed and it now becomes hydrophobic. So it originates as a hydrophilic hormone, but at the time it's activated, it's actually a hydrophobic hormone, which means that it will often rely on a carrier protein, and yes, it will favor an intracellular receptor, thereby activating transcription factors on its target cells. The other hormones in the amine groups, those all will be hydrophilic, so they'll follow the same exact pathway as we saw on our peptide slide, in that they can go ahead and freely travel through the bloodstream, and when they get to their target cell, they will interact with the transmembrane, causing a rapid non-genomic change to be initiated. Alrighty, so hopefully I was able to convey that when you want to communicate with your target cell, we need to make sure that we interact with the receptors, and these receptors can either be found on the transmembrane. This is mostly for hydrophilic messengers, and if we're looking at a hydrophobic messenger, then even though they have a choice, they tend to favor the intracellular receptors, although you do have an option of transmembrane. If you see the term ligand, that is basically another way of us saying hormones or the chemical messenger. The reason they like to use the word ligand is because the ligand is often very specific as to which receptor it can interact with. Very similar to when you discuss things like enzymes and you have a specific substrate that has to bind with the active site. And the third item on the list is that whenever you have a match between the ligand and the receptor, we will see a conformational change in the receptor, thereby allowing for activation of any type of secondary pathway or even a genomic response, depending on where your overall receptor is located. All right, so let's go ahead and take a look at some of the terminology and some of the concepts when we do ligand-receptor interactions. First off, they're very specific. So as I was mentioning to you before about the enzyme and the substrate and the active site, ligand and their receptors are very specific, so only the correctly shaped ligand will match with its proper receptor, thereby causing a response to occur. If we see that there's a ligand that looks different or it's not a match, it will be rejected by the receptor. So as you can see right here, it's being rejected, can't bind to it, and thereby no response will be generated. We also see that because there are such a wide variety of ligands, and because of the knowledge that we based on it, we can do some mimicry. So if you go ahead and you mimic a ligand because you want to activate the same receptors as the original one, then what you're doing is you're creating an agonist. So for instance, when we do hormonal treatment, if we see that the patient, for instance, is not producing enough growth hormone, but they have the receptors to respond to it, we can go ahead and synthesize growth hormone that looks just like the original one your body would make. So that would be an example of an agonist. And the agonist will be able to bind perfectly to the receptor, thereby providing the response that you are looking for. On the other hand, if you see that there's an oversecretion or you want to block some of the receptors, what you would then create is an antagonist. And an antagonist will act as a blocking agent, and it will inhibit the natural ligand from interacting with the receptor, thereby diminishing the response that's generated by that connection. So as you can see right here, it says many ligands will mimic that will, many drugs and or poisons get their effect because they'll mimic the ligand that they're trying to either upregulate or downregulate. We also see that due to the fact that we have a massive amount of DNA, we're able to generate lots of different genes and products. Some of them are the result of gene families, a collection of genes that have very similar export products that they produce. Because of this, it sometimes comes across that receptors can have what they call isoforms, binding sites that are very similar, and this will allow the receptors to be expressed on different target cells. So you can have the secretion of one hormone that can activate target cells in different organs, thereby causing different responses. So for instance, if your body is releasing high amounts of epinephrine, we see that they have target cells all over the place, but the response will be different. Epinephrine, when it's released and it makes its way over to the bronchioles, will cause vasodilation. Bronchial dilation, not vasodilation. Bronchial dilation, so that we can go ahead and we can increase the amount of air that we inhale and exhale. On the other hand, if epinephrine goes and makes its way over the intestines, usually what it tends to do is it tends to start the contraction, and it can also cause some vaso changes in the blood vessels, especially if it's trying to limit the amount of digestion that's occurring, because it's trying to conserve all the energy for you to use during your increased inhalation, and most of the time muscle use, because epinephrine tends to be secreted when we are in a stressful situation. So over here I wrote down for you that epinephrine can go over to the smooth muscles in the lungs, and it can cause dilation of the bronchioles, whereas the smooth muscles in the intestines, it will tend to constrict, so it can conserve where you utilize your energy from. We also see that we have what we like to call a law of mass action, and eventually we're going to reach our saturation point. So here on top of the PowerPoint, it says that what you want to do is you obviously want to match your ligand with your receptor, and when you do so, you create this complex. And the complex is what's going to initiate the response, because you're making a physical connection between the ligand and the receptor on your target cell. As you're doing that, what's going to end up happening is you start to get an amplifying curve, which basically says that the more you're able to connect the ligand to the receptor, the more the more amplified the response will become. So to increase the response, right over here. This is called the law of mass action. So you increase your response by keep adding or matching ligand to receptors. Now, eventually, you're going to reach your saturation mark. And your saturation mark basically says that all receptors are bound. So it doesn't matter if you have additional hormones that are floating around. Since all the receptors are bound, the response will no longer increase. It has been fully amplified. So at the bottom of the PowerPoint, I wrote down for you, receptors can become saturated, meaning that they're all full, and at that point, all responses are at maximum capacity. You cannot instigate or amplify it anymore. Another thing that we can do is that we see that cells, target cells, have the ability to down or upregulate the amount of receptors that they have. So we're not changing the number of cells, but we're changing how many receptors each cell has. And by altering it, obviously you can also thereby increase or decrease your response to it. So for instance, right over here, it says if you have more receptors, you're able to bind to more of the ligand receptor complexes, because you're able to make more matches, which means that you can upregulate the response. Now, we can also go the opposite of that, right? So over here at the bottom, it says that if you look at your target cells, within the cells that you have, you can either downregulate the receptors or you can upregulate the receptors. Downregulate means that I'm going to decrease the number of receptors that I have. And as I decrease it, it makes it harder for my ligand to bind to the receptor. Thereby, you are often saturated quicker, and you're not able to have the maximum response that you would normally have if all the receptors were present. On the other hand, we can do an upregulation. So what if we desire to have a more amplified, a more noticeable effect? Well, in that case, what we see happening is that the target cells will upregulate. So they'll increase the number of their receptors. And as they increase their number of receptors, what we see happening is that they're able to accommodate more ligand. And as a result, we see that we have a higher level of saturation. So you have a more exaggerated response than you normally would have. Please keep in mind that when you're doing your down regulation and your up regulation, that you are not changing the number of cells. They stay the same. You're simply changing how many receptors they're responding to. So for instance, when a woman is pregnant, in chapter 1, we talked about the fact that when the baby's getting ready to be born, it will require oxytocin. So what we see happen is during pregnancy, the receptors in the uterus will actually be upregulated for oxytocin, meaning that they'll be able to recognize the hormone quicker, so that go ahead and contractions can be initiated on a quicker basis. So anytime you upregulate, you just basically increase the sensitivity by allowing for more receptors to easily recognize the ligand or the hormone that you are releasing. When you downregulate, you diminish that response, making it harder for the match to occur. The other thing you want to think about, and this is what this slide introduces, is what is the affinity and what is the dissociation constant. Okay, let's start with affinity. So the affinity is basically the likelihood that the ligand and the receptor will match together. I like to think of it as a magnetic pull. If the ligand and the receptor have a high affinity, then it's almost like a strong magnet. It will quickly find each other. It will quickly bind to each other. It will have a quick response. So usually if you have a high affinity, you can get away with just producing a little bit of hormone, and you'll have a high success rate of that little bit identifying the receptors because of their draw to each other. If you have a low affinity contact, that usually means that the body will have to secrete higher levels of hormones because it has a harder chance to make that physical connection between the ligand, between the receptor cell and the ligand. I was correct the first time. Now, so affinity is the amount of attraction. And then the other one is dissociation constant. Dissociation constant means what is the concentration of messenger at which half of the receptors are bound to the ligand. Okay, so the dissociation constant takes a look at how much messenger do I have to produce so that I can get half of my receptors to be bound. Now, these two, affinity and dissociation, they correlate with each other, right? Because if you have a high affinity, that means that you can quickly locate your ligand and your receptor, which means that you will have a low dissociation constant. You will get away with producing low amounts or low concentrations of messenger, and you'll still be able to quickly mark your halfway point of receptors being bound. On the other hand, if you have a low affinity, then what we see happening is that you're going to have to secrete the messengers at higher concentrations before you can get to your dissociation constant. So a high affinity means a low dissociation constant, and a low affinity usually means a high dissociation constant. All right? One other thing to discuss is what if you have secreted your hormone, you have found your target cell, you have initiated your response, and now you're done, right? We don't need it anymore. How do we clean up? So how do we inactivate our ligand receptor complex? Well, there are lots of options. The most common option is that we utilize tissues like our liver and our kidney to get rid of the excess hormone that's floating around in the bloodstream. So this is your number one option. How do I get rid of hormone I don't need that are in excess? I go ahead and I find my liver and my kidney, and they'll degrade it and flush it out. You can also utilize other items. So for instance, we can take a look at this. It says the ligand is taken up by the adjacent cell. So as you can see, the ligand, the hormone, is endocytosed, thereby removed, so it can no longer interact with the receptor. We can see that the ligand can be degraded by enzymes. Okay, extracellular enzymes. Once again, now there's no longer a connection to be made, so no more response. The ligand receptor can actually be removed. So as you can see right here, the receptor is going to be endocytosed, engulfed into the cell, thereby removing the target. We have inactivation. Most of the time that's through phosphorylation. So they'll phosphorylate the target so it becomes deactive, and what they'll do is oftentimes they'll change the appearance of the receptor, so the hormone can no longer bind to it. And our last option is that we can activate the signal transduction pathway. So even though the hormone still binds to the receptor, we are no longer giving a response to that relationship. So lots of ways to do it, but keep in mind the number one way is to go ahead and utilize the liver and the kidney. Now within our signal transduction pathway, so this is when we're going to go ahead and amplify our response once the ligand and the receptor have made physical contact. What we see happening is that it often involves 4 components. And you've seen some of these components before. We definitely need to have what we call a receiver. The receiver is the specific part of the receptor where the ligand or the hormone will bind to. We already know that our receptor will have some conformational change, so that will be corresponding to the transducer section. And when the transducer section is activated, that means that we're able to get an intracellular response. So we're now activating the insides of the target cell. And by doing so, it will also cause an amplification to occur. Amplification means that I'm activating lots of maybe secondary messengers, thereby increasing the number of molecules that are involved. And last but not least, I will get my response. So I will get the molecular function, the changes or the secretion that I desire based on the hormone that was released. This is also one of the reasons why oftentimes if you're interested in hormones and you start studying amounts of that you have to give in case of you do like hormonal treatments or even just hormone levels in a regular healthy person, you're going to notice that you actually don't produce or secrete extreme high levels of hormones. And the reason for that is because of the fact that you have the amplification and the cascading step in the response built in. Here is a little illustrative example of our transduction pathway. So you can see that we have our ligand that's going to be binding through our extracellular membrane, and thereby it sets off a cascading mechanism that's initiated by the conformational change of the transducer, and thereby activating or amplifying, aka our cascading pathway, and getting a response that is at a much more heightened level than you would expect for a little molecule to be able to generate. One of the things that I also want to talk to you about are exactly what are these receptors look like, or what are the variations that you have when you come across them. So thus far we've discussed that the hormones can be either hydrophilic or hydrophobic, and if we're looking at a hydrophobic hormone, we see that those receptors tend to be intracellular. So that's the first type of receptors, right? Intracellular. These tend to be exclusive to hydrophobic ligands. So here on my little illustration, I could see I have my receptor on the inside of the cell. And then I have my extracellular membranes. And those are going to be 2, 3, and 4. So I have 3 options for those. My extracellular membranes are going to be for both hydrophilic and hydrophobic. However, you're going to notice that on your PowerPoint, I exclusively wrote down hydrophilic, since I've said many times to you before, your hydrophobic really prefers to go intracellular, whereas your hydrophilic has no option. It has to stick to the extracellular membranes. So when it comes to those, we have 3 options. We have a ligand-gated one, which will basically almost act like a little channel. It will allow the flow of ions or receptors or response to just go in and out. Oftentimes we see that this will lead us to membrane potential because of the ions that will flow in and out of the cell. We can have a ligand binding site. Here's our second one. Actually, my numbers are wrong. My apologies. This is number 3 on the list. This is number 2. Here was number 1. I digress. Number 3 on the list is the receptor enzyme. This one is going to go ahead and be specific for activating intracellular enzyme activities. And then our 4th option will be our G protein coupling. This will involve the gene family involved with the G proteins. And in a little bit, we'll talk about the fact that when you utilize those particular proteins, usually what you do is you either activate different ion channels or you cause a secondary messenger to be activated. Both will be utilizing an amplification pathway. So at the end of the day, it doesn't really matter if you do ligand-gated, receptor enzyme-based, or G protein-based. All of them will go ahead and do the transduction pathway, which will cause the amplification and the cascading pathway to be activated. I am going to go ahead and take a break on this slide, and I'll stop part 1 over here. And in a little bit, or whenever you're ready to, go ahead and hop on and let's continue on with chapter 3, and we'll talk about these receptors in a little bit more depth, and we'll also take a look at some hormone samples. I think this would also be a nice time for me to remind you that if you come across any questions, comments, or concern, I am available through email, and definitely utilize those discussion sessions if you prefer as well, okay? Don't be shy. Let me know if you have any questions, comments, or concern. And trust me, by the end of our third or fourth audio, I'm going to sound like a broken record, just reminding you to make sure to email me with your questions. All right, let's talk soon.